getting too. So this is the information you can go back to look at if you have time on the bus. And also this is like a very good brief introduction about the district. Because the district, our district, the goal is one is manage the water, the other one about to protect the environment, including particularly the Everglades. So this is, and I put a brochure about how about they combine the global change, the information, a little bit, very general information here, because our district also have the public relationship, so we need some information for the public. And this one is about the basic flooding control, the district's main goal, one of the main goal information. So some of them you may interested in when you go back to see how in your area about the water flood control management. This is the one. And then the other side here, this is also the overall introduction about the district, and the both as usual flooding control and the research. So have like lots of some small pictures here, and probably like this is myself like how many 16 years ago when first came from Boston, come to Florida, and they also still use what can some of you know what's this the chamber? That's like yeah, photosynthesis, major photosynthesis. Yeah, that's use LICO 62 when that's a long time ago. I don't know how about now. The chamber already changed or not. Probably the equipment changed or not. And also I know like England and European, they measure photosynthesis have different equipment too, chamber differently. Anyway, so this is another one. And I think this will be a very good, this is a very good one. And later on, you can put on the top, because later on when I talk about our STA, I will use this one as a guide. This is a very, very good brochure to talk about the STA, the generally what this we call, oh, by the way, we call the STA, this is called the Stormwater Treatment Area. So STA, in fact, is constructing wetland. So ecological engineering, the BO work, the founding father for this field. So this have the principle, and also at the back have what are our STAs here located. So this will be very good. You'll keep on the top later on when I talk about the more we'll use. And this Lake Okeechobee, you can go back, since today we'll not cover this. Yeah, this you can on the bus or go back, you can have a brief reading about the Okeechobee, the lake, how to the restoration involved. So, and the final one, this Nina one, this will be during the afternoon trip. We'll visit Nina, that we called, and I will show, when we look at the map, I will show you the roughly location today, now, and also in the afternoon, what, where. So how you have roughly location. Okay, so now, so you can keep a map and this page on the top. And when I talk, you can have the biggest, the largest demonstration in the world too, is really, really excited to talk about it, can talk about several hours to see. And also I don't talk about the Lake Okeechobee. The lake restoration is also is a major issue, is a very good, exciting too. However, I mainly talk about the Everglades in the south. The whole thing we called this one the Southern Everglades. In fact, now is the Southern Everglades. So far, the national park is down here. As I say, used to the whole this area is like like during 1800, how this South Florida is so flat, so the natural wetland. However, now you can see later since 1950, 1960, this is the area, the Everglades agriculture area, and the agriculture area mainly is plant sugar, and sugar, citrus, and some small vegetables, and like every day, like most of during the season, you see with our nearby, you see the fire. So that's the farmer burn the sugar, and so since the sugar, the plant is shifted year around. This one, So the sugar. So because the sugar, citrus, they have lots of. They use the fertilizers, the nutrients. So particularly the nutrients, particularly the phosphorus, is a major issue. So the major issue is because of the people, like in the northern Everglades, just say the water now is only have the three major water conservation areas. This is one here. This one is here. This is called Water Conservation Area 1. Is also called Arthur. What's it name? Arthur Marshall National Refuge. This is a national refuge area. And this is actually, look at here, because this is a whole area. So today our trip will cover. And this is this water conservation area. Today, this morning, we visited the place. It means STA 1 West on the northeast corner. Yeah, so here, then West 1 West. And the water, because this STA is the purpose is to treat the water flow into this water conservation area 1, mainly treated here. And on the right side, on the east side, I have STA 1 East. And this one we will not go there because now have a lot of construction. But means that both STA will treat the water get into this water conservation area 1. And then in the afternoon, you will visit the other side. So this morning we are on the northeast side. The water getting into, and you cannot see when we drive by later on. We'll see. Maybe if you have the time, we can stop. You can stand on the dike to see a little bit far away, or stand on this dike a little bit far away. See the west side, see the water conservation area 1, but cannot see very fast. However, this afternoon, you will go to drive we'll go to bus drive from basically back to the east and then to the south to reach some place here. There have like a visiting center, have very nice cyber walk for a forest, and also we have another field, like a research facility there called Lila. And the afternoon, the tour guide this afternoon, Mr. Eric Khan, he did a wonderful job to introduce to you. So anyway, so today's trip is mainly around this water conservation area 1. So in the morning we look at the construction wetland to treat the water, so get the water, good water get into water conservation area, and then later on you will see. Okay, anyway, so after water conservation area 1, we have a water conservation 2. That's the water conservation 2. So I worked for many years, work there. Like lots of field work. That is also both of them. Like I say, this is the Everglades swamp. Like it's a freshwater wetland. But these two have some differences too. Okay. Anyway, and then you see water conservation area 3. Water conservation 3 here is the biggest one. The 3. This so far have a lot of have a bigger construction and also restoration efforts there, mainly deal with hydrology. and then goes down, we'll go to Everglades National Park. I will talk, will not talk much because tomorrow you'll visit Everglades National Park. So, so this is the general one. And then, which is because phosphorus is a major issue in the district and the Everglades, because phosphorus like enrichment made our this conservation area like a cattle expansion. Like a cattle used to use minor native species, but because the phosphorus increase, they expand, replace, like for example, like a water conservation area 2 here. And almost in the first, like there is some line here, almost in the first, this line, a little bit more is covered by cattle. Used to the native vegetation like sawgrass, like water lily, all this. So we spent a lot of time, like about 20 years ago, we spent the time to study why have change, what's the change, why have the change, what's the mechanism, what's the major factors, how to conduct like over 12 years many publications, because like this research is not just like say, oh, you publish a paper. Most important is that we need to because have the lawsuit, have the state government, like a federal government, state government, and the local to have lawsuit in the court. So need to provide scientific evidence to say why made the change, what's the change, why made the change. And then use this scientific data to prove because of eutrophication, because of like a hydrological change, and then the judge made they say have to restore Everglades, have to set up the phosphorus criteria to as 10 ppb. So that's about like when we reach 10 or 12 years. And then after that, after set up the criteria for phosphorus like criteria, so we did how can we reduce? How can we do? So they came up with the STA. They came up with the two major things. One is about the best management practice called BMP, how to help the farmer for agriculture to reduce their individual farms usage, and the other ones built the constructed wetland to reduce the water. So far the oldest, because of the built up the scale will be so big. So like in early 1992, so we started the STA 1 West. During that time, it's called the Everglades Nutrient Removal Project. So this is our oldest STA. And then after their operated started 2000, and then started 2005, we have the STA 2. The STA 2. Gradually, I cannot remember. I have a table for you. Cannot remember exactly the year. But anyway, our STA 2, you can say STA 2, STA 3, STA on your right, STA 5, 6. So our is 2, 3, 4, 5, 6. So our this different. Say total we have 6 STAs to treat the water. STA 2 and 3, 4 is made to treat the water get into water conservation area 2, and 3, 4, and 5, 6 to treat the water get in conservation area 3. You probably can see we use the strategies this STA is built at the bottom of the agriculture, that when the water will directly get into water conservation area. So this is one. But now because STA, what I was told later, still is not enough. So that now district still will by land. You see this gray color. That will be future. Now some of in construction also will be increased. Will be have more expanded the construction wetland. And they also have you see that EAA reservoir. Did you see that EAA reservoir in the center? because that one is Florida. This is related about a little bit like geology. Florida is is a limestone bottom, and is a very young geological. Is like Florida is under 5,000 years, and the whole the peninsula is like 2 million years ago. So the Florida and because of flat, so like I say, they cannot hold the water. In the summer, it's in Florida subtropical, tropical season. Summer is rain season, of lots of rain and then flooding, and in winter it's dry and then drought. So like below limestone, limestone, so they cannot hold the water too. So here they say so our STA like in winter, if we meet to the dry season, really have no water. If you have no water, how can you say STA treated water? So this is proposed by this land will build the reservoir. Okay. Anyway, so like if you go to the next page. Next page have a little bit have like a more close to our 6 STA's shape. The size use pencil to write there. Like for example, the first one STA 1 West about 6,670 acres. And then like STA 1 East, our the different have the different sizes. Overall STA 3-4 is the largest, have over 16,000 acres. So this is one thing in terms of the size. Second one I'd like to point out is you look at it to see, like you see that like a white arrow is showing the water flow direction. So you see the white arrow, like each like most STS is like a flow direction. So from, for example, you look at STA 1 East, like you can see so from the north to south. So most of them so from north to south, water direction from north to south. But some of them, like STA 1 West, on the very top, 5A and 5B. One top, 5A and 5B, that's from east to west. But the rest, like cell 2A, 2B, and the rest is from north to south. Why like that? Because as I say, STA 1 West is the oldest with our pilot project. When the pilot project is not including 5A and 5B, that's smaller, and later on we add 5A, 5B. So that's the direction. Okay, this is the direction issue. Third one is I want to see can each STA, maybe I use STA 1 East as an example, is easier to say. Like for example, you can see cell 7, cell 7, the yellow, always the yellow color, the yellow, yellow green.
Yeah.
Yellow, yeah, okay. So yellow is cattle. We call the emerging plants. So because this STA originally is agriculture. So when we can purchase land, the agricultural land, just like burn all the stuff or doing some a little bit of construction work, and then we put the water in. And the cattle, emerging plants is a volunteer. Cattle because the water get in, and the land with high nutrients, they like cattle like phosphorus. So the cattle's emerging cell. So because for our STS design is want to when water flow like north to east, no matter north to east no matter north to south or east to west, always like two cells connected. So when water get into first, say emerging plants, we call the EAV. Sometimes later on you'll see this EAV, emerging plants. And then the next, so water gets into the emerging plants first, and then from emerging plants get into SAV, we call the SAV is aquatic submerging, SAV, submerging aquatic vegetation. later on you'll see all this in the field, but now you have some like idea, because sometimes I always say EAV, SAV, so you can have some idea. So as I say, EAV almost now is exclusive cattle because that's a volunteer. It's really grow very quickly. We tried to plant some other plants, really did not work. And SAV, this is submerging like SAV, so have lots of submerging plants, have like later on I can tell you like in our most species have Kara, have Laia, have different things. Okay. Anyway, so okay, let me see. Usually, so why like we think as when the water getting flow in have EAV, this cattle, and then get SAV, because the SAV is the cattle first, because like nutrients, they will remove the nutrient, they take up nutrients pretty efficient, and so they usually, like so far, we found that the cattle cell, water getting the cattle cell can reach about 75% of phosphorus removal performance. So it means if the water get into inflow water get into cattle cell is about 100 ppb, for example, and then when get into the one out from cattle cell, you see the outflow is about 35. Oh, sorry, I said wrong. I said that the outflow usually is about like 50 GC. And then this 50 GC is already relatively lower, get into the submerging cell, the next green cell. The green cell work, and then the green cell get out like average about 25 to 30. So like every time like first have like about say like 50-60% removal for cattle, and then later on have for SAV like SAV cell, the green cell also have about 50%. So overall is about like 60-70%. The removal one. You see, I use like the next page. So if someone wants to look at the data, say what the data. So this is I made the copy from. Oh, by the way, I made the copy from our 2012 South Florida ecosystem report. Because every district every year have this South Florida the ecosystem report. The book. STA have a chapter, and used to we print out, but this time they want to save the paper. They did not print out. I thought it's a print out. I want to give you a copy, but I make I had a web link, it's on the first page. Where is the very first page at the bottom?
Yeah, here.
Yeah. This, if you go back, you visit there, because they have the whole report there. This is a very, very good report. You can look for the every year's report there. That summarizes all the research, all the work the district done. It's really very informative, very impressive. Lots of information there. So anyway, okay. So now I explain this. You can see how our STA, what's the efficiency for nutrient removal. And so basically, because this is the oldest STA, the oldest. So started in 1995, and so the x-axis is the water year. Because we use a water year. So here the water year we start May 1st. So yeah, May 1st to make the water year. And then the on your right side, left side, this is FWM. That is a flow weight mean TP concentration. This is the inflow. Basically, this is the inflow TP concentration. But we did not just use the TP inflow water concentration, TP concentration. We weighted with the total flow because so this is you see inflow is if that's for the inflow is the blue bar, So you can cross to see cross this is so many years. The inflow is range is between 150 or between 100, between at least between 100 to even 300 ppb. So that's one. And the red one is the outflow TP. So you can see the outflow TP is around most of the years are around 50. Sorry, around 25, 30 average something. And then I have a few 3 years higher. So you can see overall the phosphorus removal performance, how many percent be removed. And the dash line to see how about the total phosphorus loading, total hydrological loading. Yeah. Okay. And also some other detail. Okay, and then we go to the last page. The last page is, I hope everybody to keep, I'd like to explain a little bit more detail, and then because we'll visit this place, so you'll see where where are the EAV, where are the structure one. So overall, let me see, we are at S5A. The green is here. This S5A because I have a structure, have like a pump. Oh, by the way, hold on. We need to look at the instructions legend of the map first, better. So the map, you see this for what's the color, the symbol and the color for cover, for pump. When the pump is the pump one, will have the structure there. Will have the gate there. What's the spillway? What's the wheel? And for me, actually, I'm a plant ecologist. First hour, and also from Chinese. What hour is cover? Spillway where? I have no idea. And then we look at it, see the stuff. Oh, okay. This is the cover. Oh, this is the where.
You would tell us what is the difference.
Yeah. Anyway, okay. So I will go through, but we see. Chang and I, we went to the route to say which way we show you better. You can see, we think you can see better more about STS design, STS thing, as well as can see some wildlife. But I'm not quite sure I want to see this
bus is big.
Too long.
Yeah.
I don't know.
If we cannot make, we just turn around. Yeah.
But the main thing is it will be hard. Okay.
Yeah, we can turn around. I think it's okay.
Okay. So that's so that is a limitation. I say that our dream route, and then to see we can make it. We go to the site to see mainly because relatively long. It's not wide. It's long.
Okay.
Yeah. Okay. Anyway, we are at the S5A here. And the main for this STA1 West, the main gate, the structure gate is G3. The G gate is 302.
他就要搞个跨国公司，你看。
哦，就是啊，那你就刚好在这儿来收啊。
那不过，
但是
它一个很大的问题，
这个美国同不同意啊？同不同意这个中国的这个科技型的企业？
And you all know this one is probably heard a lot of Everglades sawgrass. That is sawgrass. That's Thai top. It belongs to the sage. Over here we have like 5 vegetation type. The main purpose for this mesocosmic experiments is we just saw the STA from inflow, we drove to outflow, and the inflow we know if we come to like about average 100 ppb, and outflow is only reach like about 25, between 20 to 35 ppb. But we are required, so this is like about 36 or 75% of phosphorus removal. But we are required, the Florida law, the judge to ask we to put the water into water conservation area have to be 10. So although we did very good for SFA, we cannot reach 10. So and then also between 35, between 35, between 25, between 10, between 20. Want to this know the nutrients. Now the STS outflow, the average 25 is already very low. So but we need to reach 10. So it's under this no phosphor, this is the under 25, there's no phosphor condition. How can we lower down, further lower down? So this is a challenge. And we have district have approach called the paraphyton-based STA. It's mainly to use a shallow water, have no soil, no sediments. Dig out, have one cell at the STS 3-4, have 200 acres STA as a pilot study to took out all the sediments when built that cell, took out all the sediment, and then just put the SAV plants in and put some paraphyton to let it work. And it looks like so far this paraphyton, this system just remove the sediment. It works. It looks like can reach 10. However, you see the so big scale. If you want every well to dig out the soil, that's really it's very hard to do. So we thought about because in the nature aggregates, they mixed up with two major vegetation types. One is this sawgrass, with this one. Just when you touch, be careful. The sawgrass, the edge really have the thorn. So if you want to from lower to up, it's okay. You cannot from up to down. Okay? If you say. One is the sawgrass. The other one is water lily. If you will say water lily with Eliacus and the paraphyton. These two major type of vegetation type grow in the natural habitat with 10 ppb. So that's say so why they can based on our study, preliminary study in the past over 10 years, we know sawgrass have pretty good, can grow in the low nutrient condition, have higher phosphatase enzyme, and can store nutrients in the below ground. And the same thing we think for water lily too. So here we'd like to compare 5 vegetation types grow under the very low nutrient condition. The water source of this plant, the experiment take from the canal. This is the canal. So the outflow of the SUK west we just breathe and will get into water conservation area 1. So we take that from that canal to come to this the pipe connected. This is the central tank, the central. And this is like the range is about 25 to 35. And then we plant our this vegetation like 2 years ago with different vegetation type. For sawgrass, we only put like per 12 small plants per square meter. Then for cattle there, we put it like about 12 small cattle there. For the SAV here, we can see 2 types of SAV. And the one is the Kara. Like this one is the Kara. Maybe you can see the yeah, you can take out. So this is the Kara. The Kara is a algae. It's algae. They have no roots. Yeah, tiny. Yeah, no roots. It's a large algae. They take up the nutrients from water column. And also, Kara is very good to take up the combine. You see this little reddish thing that's combined with carbon, the calcium, carbon calcium. They will combine together. This we have this one Kara, and have another one's Miao. We grow them, and this is a 2, and also sawgrass cattle and water lily. So we measure the water quality and the redox, temperature, and measure the plant activity, measure the soil nutrients, and doing our and when they have 1 year, we also measure enzyme to doing a sort of the things. And the water, particularly this is a tank, is about 6 meters long. And so every tank you see have this one's inflow side. The inflow side, Chang, actually you should talk. You should talk water system. You talk.
Barrel.
Yeah.
It's, so let me talk about how the water get into the tank.
Yeah.
So we have 2 timer to control the water, the inflow water get into the tank. The first timer they set up to open the pipe to get the valve, let the water get into the tank, the barrel, blue barrel. And the timer number 2 they set up. You see the switch. You can see it here. You can see this is switch to let the water from the barrel the tank into the low barrel into the tank, individual tank. So we have 2 timers to control the water, inflow water get into the tank. And each day we have 40 gallons. So that is the residents. 40 gallons get into the tank. That is what is we can say it. That
Yeah,
is,
that's we calculated the volume of the viscous, and then we say the 45 years ago when we plant water lily for the based on our square meter. Also we plant Ediacus. We took Ediacus from here. So this is the Ediacus source of our other tank. This one, in this tank has no soil. Our irrigated water is from rain. So this stays there for over 10 years. You see, this is just like this spot. This is weed, the same. We just grab like there like spots like this. You see the Ediacus, they also have below ground. This is the Ediacus roots, and they have below ground rhizome, but the rhizome very short, very short. This is the rhizome, very short. So this is how we grab like under 12. We grab under 12. This Ediacus too. So the water lily is very interesting since say. This one is a white flower. It's different from the one we just saw, the yellow flower. Yeah, yellow one. The other one is when we first put water lily here, it really has a lot of the flamingos. They're caterpillar. Let me see how the caterpillar eating the water lily leaves.
Yeah, look at that, like a hole.
Yes, like you see here, for example. See this small piece? I know this small piece under this. I have a worm. So this in spring, this worm very a lot. So that's eating. They eat the leaves. This leaf piece also touch here, but probably eat from.
there is a flower over there.
Yeah. So when we
Flower.
put it here, maybe because we introduce a new population here, this worm very happy, just flew away. They spend like before a lot of postdocs from Korea, South Korea, spend a lot of days to every day we just split. So okay, this is one thing. The other one is water lily, like also year round, but in winter, relatively grow slow. And this also in this, before I never noticed the water lily have 2 types of the leaves. One is this big leaf. The big leaf, we can see this is the leaf will come up. I can see this big leaf is a big leaf. But I also have a tiny, smaller leaf. This tiny leaf is just different type. When they grow up, they're still like this small. Yeah. So if here, I can taste inside still have. So, yeah. So we just. Yeah.
I work here at the refuge, but and this is a federal facility owned by the United States government. But I work for the state of Florida on a project called Lila, and we'll talk a little bit about that. We might take a walk down the levee and kind of have a look at it. But I was going to start out and tell you guys a little bit about the wildlife refuge and the refuge system in general. It's generally known as kind of the world's model for protecting natural lands. There's wildlife refuges across our whole country. There's over 500 refuges. There's one in every state, at least one in every state. So we're down here at Loxahatchee. One of the very first wildlife refuges was dedicated by Teddy Roosevelt in the 20s, I believe. And that was here on Pelican Island. And that was to protect wading birds and waterfowl from being overhunted. I mean, that system has spread throughout the whole country. A lot of these wildlife refuges were, the purchase of that land was funded through hunting, through taxes on hunters and permits to hunt. So we have a long history of that. What's sort of unique about this wildlife refuge, and here's kind of an overview of, you've got that on your map I gave you, is that it's called an overlay, meaning a state or another entity owns the land, but the federal government manages it. And we're one of the few refuges that work like that in the country. So my agency technically owns the land, and we lease it to the federal government, and they manage it as a wildlife refuge. It was created in the 1950s as part of a series of impoundments where we kind of created these canals around, what was left of the Everglades, a series of canals and levees. And we call them water conservation areas. They were meant to hold on to water, primarily for flood protection and consumptive use. When we went into this process of creating these water conservation areas, there's 3 of them. The refuge is about 150,000 acres, and then there's one that's about 300,000 acres, and another one that's 150,000 acres. It was decided that we would manage one of these for wildlife. And that turned into the Arthur Marshall of Loxahatchee National Wildlife Refuge. So it was established in the 50s. And like I said, the state of Florida actually owns the land. Now, the land that we're standing on is part of a series of compartments that the federal government actually purchased in fee title. And they did that to manage for wintering waterfowl habitat. So for many decades, there was a series of impoundments and compartments that were managed to provide habitat for waterfowl to winter here. And then they had a series of what they call quota hunts, and they would hunt waterfowl here in the winter. And they did all sorts of manipulations to create really great habitat for waterfowl in the 50s and 60s and the early 1970s. At that point, sort of the focus was shifted in trying to manage the compartment systems for a more natural suite of birds and wildlife. And that was sort of the backdrop for this Lila project. So we got started construction in the year 2000, and it took about 2 years to completely construct this system. Basically, we took 2 existing impoundments and we made 4. And what these are replicates in their physical model of the Everglades. So each one of them contains sort of these 3 key habitats that you would find in the Everglades system, and we manipulate the water levels in there to elicit a restoration response. And I have a series of studies going on there, and different researchers from all over the world testing restoration strategies within this model. And for example, we study tree islands, which is a really important part of the Everglades natural landscape. It's the highest part. And almost every animal that makes use of the Everglades, at some part, its life cycle is dependent on a tree island. And we have ridges, which is like just sawgrass, and then we have open water sloughs. That's the lowest part of the Everglades. And so my job is to control those water levels and try and keep my mad scientists happy that are testing restoration strategies out in Lila. And so what we kind of look at is that response between a hydrology, what we think of as a restored hydrology, and tree islands. So how do you need to manage the water in order to restore tree islands? Because out in the natural system, we've lost anywhere from 40 to 90% of the tree islands out in the Everglades. We manage the water to elicit a response from wading birds, long-legged waterfowl, because we lost anywhere from 70 to 90% of the wading birds that once made use of that system. And we know that a big part of that is the fact that the birds have timed their breeding cycle to the natural hydrology, in particular the highs and the lows of the rainy season, where we're at the peak of the high water right now, or the low water in the very late spring, early summer. And so we have, different suites of scientists that are looking at, they look at from the top down, they look at wading bird behavior and how that's influenced by water level. And we also, from the bottom up, we're looking at wading bird food, essentially small marsh fish and crayfish, decapods, and how their population responds to that change in hydrology. And so we have 4 replicates, and 2 of them we maintain a flow through, a constant flow through, and 2 of them are stagnant. 2 of them the water doesn't flow. But I try and keep the water levels the same across all 4 in terms of water depth, how deep the water is. So for an example, and we'll take a walk, it's just right down the road here a little bit. One of the most bold studies that we've attempted so far is we recreated strong drought conditions in 2 of the cells, and 2 we left on this sort of normal seasonal pattern of hydrology. And there's this theory out there that in the Everglades, crayfish, does anyone know what a crayfish is? It's like a little insect, kind of looks like a little lobster. That they respond very positively to drought. Because when the water level drops, the fish obviously have to go with the water. That's the main predator for the crayfish. But crayfish can burrow and estivate in the mud, and they wait for the water levels to come back. So when the water levels come back, the crayfish come out of this burrow in a predator-free environment. And so their population goes through the roof. It responds very positively, and the birds take advantage of that. So if you think about trying to experimentally tease apart how that might work in 150,000 acres and 500,000 acres of what's left of the Everglades, you can't do it. It's impossible, right? But on a smaller scale, but still a landscape scale, across 80 acres, we have that power to manipulate water depth and create those drought conditions. And what we saw was a very strong support for that hypothesis, that within 40 days we saw a huge increase increase in the number of crayfish in these two manipulated cells versus our control cells. And that's a 3-year study that actually was finished up this last summer. And every year we see a strong response from the wading birds. Is not very representative of the Everglades. You're looking at the effects of this canal and levee. It should be a water lily slough, dotted with some tree islands and open water and sawgrass. What you're seeing is cattail and willow. Alligator?
We haven't seen
You
alligator
guys didn't
yet.
see one?
Yeah.
Oh, we gotta come down. There's
Yeah.
one right there, a little guy.
Okay. Because I emailed those guys, we will see gators. So far we haven't seen it.
Well, what happens here? We have a lot of people fish,
Yeah.
and the alligators have learned to steal the fish off the line.
Oh.
So they're drawn to people. Did you see any down here? Was there any down here? No. There, like I said, there's one right there. I can see his eyes poking up.
Oh yeah.
You see?
Yeah, the eyes, the head.
We can walk down there.
Yeah,
if
we
you want.
can.
He might duck under the water if we get too close. He was down here. He was like, I mean, his neck had to be that big around.
Wow. We will see tomorrow. We are going to.
Going to the park?
Yeah.
Yeah, you'll see some tomorrow for
Yeah.
sure.
Sometimes there are lots of in the STA size,
Oh.
tons of them. But
Yeah.
we
What
don't
did you
see
guys
today.
see at the STAs? You see anything?
No, and just plants. You see the gators?
You did?
Oh, good. Oh, good.
Yeah, for the
Okay,
first
good.
time next year, we're going to allow alligator hunting in the refuge.
Oh, really?
Yeah.
I better come. You have a license?
You have to have a license, yeah.
They have a python here?
No pythons yet,
Oh, okay.
but we expect them.
Oh, you
Yeah.
expect them?
Yeah, well, there was a big cold snap in 2010
Yeah.
that pushed them way south.
Oh, okay.
So they would probably be here by now if it wasn't for that.
Oh, in many restaurants we can enjoy crayfish. However, most of them are natural or just stocked or
Natural.
natural. Oh, I see.
But it's actually that species of crayfish
Uh-huh.
that's become a problem worldwide as an exotic crayfish,
Right.
especially in Europe.
In Japan too.
In Japan,
we have
too.
lots of American crayfish. Yes.
Yeah. It's funny, and here we're trying to manage for crayfish.
Uh-huh. Nobody tried to eat crayfish in Japan except me.
Right.
When people will be hunting for the alligators, what kind of methods would they use?
But primarily they use like a fishing line and a fishing hook, and then you use what's called a bang stick. It's like it's a gun that only holds one bullet, and it's on the end of like a pole.
Uh-huh.
And you discharge it underwater, so it's pretty safe. There's no ricochet, and it's very quiet. And
Alligator meat is so common here too.
so it's important probably to differentiate between what we're doing and the STAs. The STAs are dealing with water quality. And this project is interested in studying the other side of sort of the water issue of the Everglades. We want to understand more about water flow, water depth, and hydroperiod, or the amount of time that a certain habitat is flooded. So we're working on sort of the other side of the equation. My agency has a sort of mantra. It's called getting the water right. And there's sort of these 4 main problems with the water and how it's managed currently. And so my agency, the state of Florida, managed that water quality issue with stormwater treatment areas and best management practices in agriculture. as far as I know, we are the only site that's really studying the other parts of that water issue, that timing of water, water depth, and water flow. And so what we have a 125 horsepower pump. It's kind of up where you can see that roof over there, and it pumps water up through this cell and into these water control structures right here, and you can kind of see there's 4 as you go down. And the water level and the water flows in through those. There's a 36-inch pipe there. And it flows into that pipe across the model, is collected in a canal down there, and it moves in a big circle. And so every 15, my joke is, when if this translates very well, is that this is probably the most highly monitored and managed 80 acres in the world. And that's not that much of a joke, really. Every 15 minutes, I figured it out once, we generate, just on the hydrology, we generate around 2,000 data points a day across this 80 acres. So we have a really good idea of water depth and how much water is flowing into and out of the model. And it all gets stored. There's a data logger in that control box there, then it goes up wirelessly and it goes in real time into a database. And it goes out on the internet, so anywhere in the world you guys can look back and see what the water conditions are at Lila, although I'm pretty sure I'm the only one that actually does that. Maybe my boss on occasion to see if I'm doing what I said I was going to do. But so you can see our tree islands. We planted those trees in 2005. We planted about 6,000 trees across our 8 tree islands. And now, basically, we're trying to kill them with water. And we want to find out where along the slope of a tree island is the ideal hydrologic conditions for any particular tree species. So every 6 months we do 4 measurements per tree. We do tree height, crown width, crown length, and height to the lowest branch.
Okay, for those trees, did you planting or just?
We planted them. Yes.
Oh, okay.
Yeah. We planted them in this set in 2005, and then the middle 2 we planted in 2006.
You choose both cypress trees there?
No cypress trees.
Yeah, you
Far
should.
away from here, you can see both cypress trees.
Yep, those are now probably historically the tree islands in the northern Everglades did not have cypress.
Oh, I see.
Because cypress likes deeper water than a tree island. Now we do have some in the southern end of the system, and you guys will see those at the park for sure tomorrow.
I see.
Here it's more of a hardwood species. One of these we have is red maple, which is kind of a cool tree. It's rubrum. This is about the southern end of its range, but the northern edge is the Arctic Circle. So it covers that whole, almost the entire North America and beyond. It actually does very well here. In fact, it's some of our taller trees. We have a strangler fig, a Ficus aria. That's like this one you see on the corner is a strangler fig. So that's my record growth rate in 6 months. We had a ficus grow 1.5 meters in 6 months.
What species of ficus?
Ficus aria, strangler fig. And so all we have out there is a native suite of hardwood species you would find in the northern Everglades. And that's important distinction, really, because we're kind of crossing a boundary for a lot of tree species where you guys are at right now and where you'll be tomorrow. And so the Everglades is sort of broken up like that in the northern and southern Everglades. We look at flow impacts. One of the cool studies that we've done, you can see these boardwalks that kind of come out into the open water. We have these guys from Scotland build us some fake dirt.
Fake dirt.
Fake dirt. And the reason they did that is, you know, sediments are moved around in the Everglades. We assume. If you fly over the Everglades, clearly it looks like it's shaped by moving water. It looks fluvial. But there doesn't seem to be enough energy to actually pick particles up and move them from the slough to the ridge, which is what we assume had to happen to create that microtopography. But, you know, all this manipulation and alteration of the system happened long before we could measure the flow rate of water. And so what we did at Lila was we put that fake dirt in the bottom of the slough, and then we cranked up the flow rate, and we looked at, okay, when are we seeing suspension of that tracer particle? And it was kind of a neat setup that the particle fluoresced, so you could detect it with fluorescence. It was also magnetic. So we suspended these like 11,000 Gauss magnets off these boardwalks, and as the particle would flow by, they'd get stuck to the magnet. And you get this sort of, when you measure the mass change of the magnet, you get this sort of 3D image of how the soil particles moved in relationship to the speed that water was flowing past. And that was a neat study, and we changed a lot of people's mind about how fast historically the Everglades probably had to flow in order to create that suspension. You know, there's been a lot of numbers thrown around, but people have gone down as low as 1 centimeter per second. Well, we know that now that that's too slow to move particles around in the Everglades. It's probably closer to 3 centimeters per second, which has a big influence on restoration. If we decide to manage for 3 centimeters per second, you have to have a massive amount of head upstream to create that flow, to go from what's left of the Everglades, the northern end to the southern end. So that was a neat study, and it's not hard to convince guys from Scotland to come here and do field work in the winter in South Florida. Because it's pretty cold there, and it's really nice here in the winter.